So hello everyone and welcome back to SID2004. We'll just continue what we left from last week's lecture. Okay, so in general what we've looked at is we looked at the primary structure of a protein, and we've also looked at the secondary structure or secondary levels of protein. So primary is just a sequence, and secondary is basically on how the sequence fold on itself to form either one of the three secondary structures that were covered, so which are alpha helices, beta sheet, and the random coil. So today we're going to look at the tertiary structure and quaternary structure, followed by a little bit on protein functions and diseases, and we will see where we are heading. Okay, and as a tertiary structure, as I mentioned, it's just need to be in the same polypeptide chain. Okay, so if you look at the blue, it starts from the C terminal here, ends at an N terminal on the top over here, and they are all connected via the same covalent backbone. Okay, so what makes it different is that in the tertiary structure, you do have multiple secondary structure. Over here, for example, over here you have a beta sheet. Okay, and over here you have the alpha helix, or simply just helix. Okay, and you have a random coil everywhere else. And when you have a mix of secondary structure like this is what you call as the tertiary structure. Okay, as simple as that. And this is just to show you that within a tertiary structure, you do have multiple types of forces that keeps the tertiary structure intact. And the random coil, as an example, you can see here that it forms, what is shown here is that it forms a certain shape. So this shape, which is not alpha helix or beta sheet, this is what we call as a random coil. And this random coil are held by strong forces such as that of a bridge or electrostatic interaction. So there are rather not really fixed. Okay, so like alpha helix and beta sheet, the bonds are very strong, but for random coil, due to just electrostatic interaction, they might not be as strong as alpha helix or a beta sheet. Okay, so a tertiary structure of a protein is a three-dimensional arrangement of all atoms in the protein. So all atoms. Okay, so that's the keyword. Protein or polypeptide. So this is a very critical thing to do, to say, because some protein, they are made of multiple polypeptides. So the proteins are folded simultaneously in a solution to maximize their stability. So there is actually a concept of spontaneous formation of a tertiary structure. If you go in a very detail, it's actually unique in the sense that researcher has no idea on what promotes the folding. So imagine if you have one amide bond can have two different angles by which they can rotate to a certain degree. Okay, so that one can have a certain rotation, and that bond can also have a certain rotation. So just simply by thinking about two amino acids, you can have different degrees of rotation. Imagine if you have 100 amino acid protein, so you can have thousands or probably millions of degrees of rotation. So how does a protein rotate in a very specific manner? We know the basic concept. We know that alpha helices and all the other forces that are involved in the formation of the structure, but we don't know why it turns a certain way. Okay, because imagine we only have like 20 naturally occurring amino acid, but how can we make up so many different types of proteins with so many different functions? Okay, so that's why it says here spontaneous but because we don't really know basically, but there are research groups that are trying to figure out on why does this happens. Okay, so stabilizing interaction in a protein can occur between peptide groups, side chain, and peptide and side chain. So this is also, as I mentioned, if you look at the previous slide over here. Okay, so this is the hydrogen bond between the R group and the backbone. Okay, just an example. This one is between two R groups, and of course on a beta sheet and alpha helix, you know that it's from backbone to backbone. Okay, so these are just some example of stabilizing interaction. So more about tertiary structure. The most important factors maintaining tertiary structure is hydrogen bond, the covalent bond that has roughly about 7 to 40 kilojoule, electrostatic interaction, non-covalence, polar residues from the side chain has about 20 kilojoule per mole. So it's in the middle between hydrogen bond. Van der Waals, you have roughly about 2 kilojoule per mole. Disulfide bond, so it's a covalent bond, so it's very very strong, about 250 kilojoule per mole, and the backbone structure, which has roughly about 150 to 400 kilojoule per mole. So yeah, so these factors are the forces. These forces holds the tertiary structure intact. Therefore, there might be some flexibility. If you look at this, there are always a range, so there are a little bit of flexibility between the forces. Thus allows for a little bit of movement for the protein. Okay, just to quickly go to see some examples. Hydrogen bond, non-covalent bond strengths, as I mentioned previously, forms between a proton or hydrogen to electrostatic. So those are the examples of the R groups, and an example shown in this figure over here is between an asparagine and serine amino acid. So um, asparagine over here and serine over here. Okay, so it forms a hydrogen bond in the middle there. Similarly, for electrostatic interaction, so a different charge, the bond strength is about 20 kilojoule per mole, formed between the opposite charge or polarity, lysine and arginine plus forms a cation, while glutamic acid and aspartic acid normally forms the anion. Okay, so and shown here, this is lysine and the other one is aspartic acid. Q is glutamine or D. Alright. Another example of electrostatics or hydrophobic or van der Waals forms between two nonpolar R groups. So again, this is just a revision. So the amino acid is basically all amino acid that does not have polar or charged groups. An example here is also shown from the insulin is leucine and leucine. Okay. Oops. And finally, there's of a bond, one of the strongest bonds in the in any peptide that contains two different cysteines. So the bond strength is about 250 kilojoule per mole, formed when oxidation of too close proximity of cysteine occur. So must have two cysteines. If you don't have two cysteine, then it doesn't form. So again, this one is shown from insulin chain A, disulfide bond in red. So it greatly stabilizes protein tertiary structure, but it is less abundant in protein structure because cysteine is involved in bond formation. Okay, so just a little bit of chemistry about disulfide bonds. So mild oxidation joins the two thiol group from cysteine to form a disulfide bond.
And of course, disulfide bonds are reversible. So if you reduce it using a reducing agent, you can break the disulfide bond, forming two thiol group again. Okay. Again, this is just an example of disulfide bond from insulin. Okay. Yep. So that is the structure of insulin enfolded. So chain A, remember initially when I started showing this lecture slide, you have two chain for insulin, chain A and chain B. So chain B is in cyan and chain A is in green, and you can see that there are three different disulfide bonds that interconnect between these two chains. Okay, so just to show you here, so this is what allows insulin to have a very solid structure, and it is functional, but it is still breakable and recyclable. Okay, some proteins have two or more polypeptide chain subunit as of insulin. So a protein with a single polypeptide chain, a subunit called a monomer, two subunit forms a dimer, three subunits called trimer, and so on and so forth. So four subunits called quaternary. Right, so quaternary structure of protein describes the way the subunits are arranged with respect to one another. The subunits of a protein are held together by chemical interaction, hydrophobic interaction, hydrogen bonding, electrostatic interaction, in a three-dimensional conformation. Okay, so this is another example, insulin, by which the interconnecting bonds are the disulfide bond. However, if you looked at hemoglobin, so the protein molecule that carries oxygen and carbon dioxide in our red blood cells, so it is made up of four different polypeptide subunits. Okay, so you have two of alpha chain, you can see here alpha chain, and two beta chain. So four of them forms the hemoglobin protein, and hemoglobin proteins carry our oxygen throughout our body. Okay.
Alright, so to the next stage, protein biological function and disease. So, as I've mentioned, the quaternary structure or tertiary structure of a protein actually require for a protein to function properly. Okay, so each protein, regardless of what their biological functions are, so it can either be in a form of an antibody. Okay, this is just an example. Okay, you can have transport protein, or you might have something like an enzyme. Okay, so these are all categories of proteins. It has, of course, it has a different function. So a transport protein, for example, function as to transport molecule across membrane, or from one place to from one location to another location. Okay, so antibody protects us from diseases, viruses, and so on and so forth. And enzymes, there's a lot of different functions of enzymes. So some enzymes degrade molecules, some enzymes promote the formation of molecules. Okay, so protein with similar function may have different sequence and structure. So something that should be straightforward. As an example, hemoglobin in the red blood cells carries oxygen in the form of a tetramer subunit. So what does it mean? So it means that you have one hemoglobin, so you need four of them to actually function properly. Okay, so in other words, myoglobin, the oxygen carrier protein in human muscle, is a monomeric. Okay, so this is so to say that even protein that function similarly might have different sequence altogether, and might need a different environment for it to work properly. Okay, so hemoglobin, you need 4, so 4 subunits to function. But myoglobin, so these two has the same function, to carry oxygen. Okay, but myoglobin is found in your muscles, hemoglobin is found in the blood. Okay, but even if the functions are similar, but the structure of those proteins are different. So something like this can be considered as a tertiary structure, while a tetramer is a quaternary structure. Okay, so having said that, protein structure is unique to its own function, and therefore unnatural modification will result in lack of function. Okay, so unnatural modification. So we will look at some example of diseases that occurs as a result of unnatural modification. Okay, so one example is sickle cell anemia. So we know that our red blood cells, normal red blood cells, are normally look like this on the right hand side. Okay, it's kind of like an infinity shape. So or a double, how can I say that? It's an elongated in one way, all right? But in the patient with sickle cell anemia disease, the shape of the red blood cells forms a sickle. Okay, a sickle. So for those who don't know what a sickle is like a sabit. You know, in Malay we have this sabit where you use a sickle to clean out a land from grass and whatnot. Okay, due to a substitution of one amino acid in the hemoglobin protein. So it's a very simple modification. However, the structural differences are like heaven and earth, so to say. Okay, so this one amino acid substitution changes the way protein folds. Similarly, okay, if you were talking about COVID-19, so you might have heard about single mutation or double mutation. If you actually read through the mutation are actually either a single amino acid substitution, or in case for the Indian variant, it's a double mutation. So there are 2 amino acids substituted. Okay, so it's a very simple substitution. However, the changes are very huge. Right, so similarly there are a lot of other diseases that are as a result of protein malfunction. For example, diabetes, cystic fibrosis, Alexander disease, Alzheimer disease, or prion disease. This is just to as a list for you guys, but just get to know of the disease. I'm not gonna ask in the exam, but it's good to know about these diseases to just increase your knowledge. Okay, all right, so additionally what might happen, so in one way, if you change the structure of a protein in a natural way, it might result in different function. But what might also happen is a protein denaturation. So what it means is that normally in the normal levels of proteins, so you will see primary forms a secondary, forms a tertiary, forms a quaternary, or its function. Okay, of course the tertiary can also have its own function. Okay, but what denaturation does is actually the opposite. So if you have a fully functional protein, if it denature, it can become a tertiary. Of course, it depends on the degree of denaturation. So it can form either a tertiary structure or a secondary structure, or it might even denature or change the protein from a 3D shape model into just a simple primary structure with a long chain of polypeptide. Okay, a very common example is boiling an egg. Okay, so if you boil an egg, what you will see is normally the egg is of course the middle one, the yolk is always yellow. Sorry, the yolk is the one which are normally clear. Okay, but once you cook it, what will happen? Well, the clear yolk now becomes white. Okay, so this is one example, a very common example of protein denaturation. Okay, so what it says here is what happened is actually a destruction of the unique three-dimensional folding of a protein. So imagine inside here, initially the protein folds into a very tiny dots. Okay, so that's why light can pass through at least a certain bit, so it becomes like clear or cloudy a little bit. But once it denatures, what happens is that all this protein becomes a linear and kind of like fibrous. Therefore, light can no longer pass through, thus it becomes white. Okay, so of course it reflects. Right, so it leads to temporary or permanent loss of function or activity. So an egg, a boiled egg or a cooked egg is a total loss of function. Of course, some scientists actually did investigation if you can actually turn a boiled egg into a clear egg again. And it's very amazing. The answer is yes, you can, but it requires a lot of energy and so on and so forth to actually convert it. So yeah, it's kind of like interesting. So it's like unfolding the protein into a linear form, thus changing its physical-chemical properties, as I mentioned over here. Okay. All right. So how can you denature protein? So basically, there are three main ways by which a denaturation can occur. Alright, so the first one is pH change. So as you know, certain amino acids has its own charges, right? So and based on the tertiary structure slide that I've shown you at the beginning of the lecture, what you see was that there are a lot of function that plays a critical role in holding the structure intact. Okay, so if you change the pH, meaning that you are changing the charge of many residues or many amino acids, thus causing changes in electrostatic interaction and hydrogen bonding, therefore kind of like break down the structural motif. Okay, so pH change is one example of how denaturation can occur. Second one, chemical additive. Again, similar idea, whereby if you add in chemical additive such as urea, guanidine, or you just use an organic solvent, what it can do is basically it can substitute the charges or interaction that are occurring within the protein itself. Thus, it might form a strong hydrogen bond with the protein groups and therefore kind of like denature or destructuring the whole three-dimensional structure of a protein. Okay. Additionally, heat or agitation. So this is an example of how if you're boiling an egg. So it increases molecular motion by which it disturbs or destroy the forces holding the protein structure in place. So if you boil an egg, you destroy the structure, therefore it becomes white. All right. So a summary for this section is basically protein functions are highly dependent on its structure. Structural malformation that occurs naturally forms a disease. In some cases, it can also lead to death. But structural deformation that occurs unnaturally, like cooking and whatnot, might not be causing death, but you still need to cook it to eat, right? And induced deformity is possible by breaking the forces that holds protein structure together. And an example that you can what you can do to denature is by changing the pH, changing the temperature, changing the solvent, induce agitation, and so on and so forth. Okay, so as I mentioned in the previous slide. All right. Now, we've looked at the structure of proteins. We looked at a little bit on very basic on the function of protein. We also look at on how denaturation of protein can occur. So let's go to the chemistry bit, which is chemical synthesis of proteins and peptides. Okay, so protein and peptide synthesis can occur via conventional synthesis. What do I mean by that is like when you are doing your organic 1 or 2 or 3 laboratory, whereby if you were to produce something, you'll just take a round bottom flask, put a stirrer bar in there, and then just put the reagents that you want. Okay, sometimes in equal manner, sometimes in not in equal manner, depending on the reaction. Okay, so what you can do the same thing for protein and peptide synthesis. So you just combine an amino acid, say for example alanine, and then amino acid B, glycine. Okay, you can just put it into the same raw bottom flask, stir it, and after a while, voila, you will get a combination of alanine and glycine. Okay, so basic principle. So say if you want to synthesize a dipeptide, the barrel is glycine and LR. So as I mentioned, you can just combine those two together. However, what you will get is not just simply glycine and LR. Okay, so some side reactions will occur. For example, of course, you will get your glycine and alanine dipeptide, but you might also get alanine and glycine dipeptide. You might get glycine and glycine, and you might also get alanine and alanine. Alright, so these are the four basic possibilities that you can get. Of course, if it's just a simple like this, okay, you can add, basically you can get an unlimited combination of synthesis. You can get a triple glycine, a triple alanine, or you might get a six-tuple glycine, and so on and so forth. Because there is no limitation, what you have here is just a basic amine group and a carboxyl group, and therefore you can have either glycine with an free amine group connected to alanine via amide bond in the middle there, and then you still have the carboxyl acid group as a free carboxyl acid group. So that's why I said you can basically have a multiple combination, because you still have the active functional group. So even if you have two of these, okay, even if you have two of these, so you can still have this glycine, the amine group from the glycine to attack the carboxyl terminal of the alanine. Therefore you will get GA. Okay, so it's a lot of combination. So how do you actually synthesize them? Okay, so your four products are just the bare minimum. You can actually get unlimited combination of protein and peptides. Okay, so so that's why I said polymeric reaction. Okay, so what are the strategies that you can actually use to control the peptide formation or the protein formation? So if you divide this into two sections, so one section, the conventional way, which is liquid phase synthesis. So basically it's just the normal way by which you just combine two things in a solvent and then you'll get your product. Okay, the first strategy, what you can do is to introduce a protecting group. Okay, so if I'm not mistaken, you actually did learn a lot of ways by which you can introduce protecting group. Okay, for example, a simple protecting group for a ketone or an aldehyde is by introducing a diol. Okay, so this is just an example. So these are the protecting group, and towards the end of the day, what you will have is you will have a ether link like that, and then you can perform your reaction, and then finally you can convert this back into aldehyde. So similarly so this is the basic function for organic protecting group. Okay, similarly for peptides and proteins, what you can have is what you can introduce is you can actually introduce a protecting group either on the N terminal, or you can introduce protecting group on the carboxyl terminal. So you can have either or protecting group on either side of the protein, and then And then what you will have is that you have an amino acid which are protected on the N terminal, and then amino acid which are protected at the C terminal. And once you combine them together, what you'll get is you'll get a dipeptide with a protected N and C terminal. So if you have something like this, then you are for sure to get a single amino acid, a single dipeptide combination, and you won't get any other combination. Okay, so in case if for example this one is alanine and that one is glycine, therefore the only combination that you will get is alanine and glycine. Okay, because you have two protecting groups on either side of the dipeptide. Okay, all right. So of course there are a lot of other strategies. So removal of all protecting groups, removal of N protecting group, and you can also remove of the C protecting group. So depending on what you want to do next, so if say you already have your dipeptide over there, okay, if you remove both protecting group, so you can probably use the dipeptide straight away for whatever reason. If you just remove the N protecting group, you can couple another amino acid, and then you can actually grow the peptide at the N terminal. In contrast, if you remove the protecting group on the C terminal, you will grow the peptide towards the carboxyl terminal. Okay. However, the growing of peptides on the C terminal are not favorable because it might introduce racemization. Okay, it doesn't mean that it is not working. It does work, but it will introduce racemization. But interesting thing about this is that these are how nature produce peptides and protein. Okay, so it's very interesting. Why? Of course, again, because nature utilizes enzymes which are highly specific to either one molecule or one function, and so on and so forth. So for us to mimic this C terminal growing of peptide is possible, but you might need to use a chiral ligand or something like that. Okay. However, the N terminal modification or N terminal path of synthesis actually works better, and we will see some example in the next few slides. Okay, so the first strategy, protecting group. So protection can be carried out on the this is just a summary on the amine group or the carboxyl group. So carboxyl group, the normal protecting group that people use is esterification. At the N terminal is acylation. Okay, because you have these two molecular function. One is basic and one is acidic. Therefore, you can introduce molecules that has both protecting groups or either one of the protecting groups. Okay. And we will just continue the lecture soon.